Resources Institute Information Network or something like that. And basically, if you just Google that, you can get on this network. And Frank is an expert at this. But basically, you can select seed of just about any crop in the world that comes from all over. You'll get very small amounts of seed. And it's meant to help catalog seeds so that plant breeders, researchers, and farmers can get this seed. You get very small amounts. Actually, in the Einkorn, Emmers, and Spelts, we got 1 gram. And I want you to remember that story now, the amount, as I go through this, some things here. You get very small amounts of seed. But what we did is we evaluated these then to try to, with the idea that we're going to eventually, by the time the project's done, which started in 2012, we'll finish in 2015, from this 1 gram of seed, we're hoping to see it planted on field scale by a farmer or farmers. And I'll have some scenarios to show you how it's working and stuff as we go through that. But that's a key point I want you to remember, that this can be done by anybody. And with much of this seed now being very rare and somewhat valuable. So this is what we started off with. Now if we look at this slide here, this is actually in 2012, the first evaluation. And this happens to be Emmer. And there's 117 of those. Now those rows are only 4 feet long. So that's what we started off with, that 1 gram of seed, or those 30 seeds to plant those 4-foot rows. So with Emmer, we had 117 of them. And with Einkorn, we had 97. Now we were selecting more than that, but we didn't get them all, meaning they were out or they didn't have seed. And that's why when you look to Spelt, and we are working with spring crops, I should mention also, these are all spring, not winters at this point in time. But with Spelt, we only got 67. Our target goal from the grant was to say we wanted to do 100 of each. That's what we ended up with when we planned that first year. So if we look at that, it's a little bit harder to read, but that's what we're evaluating on those. And what we want to do is evaluate those and increase them and to get to field scale. Now we're whittling them down. So if you look at it, the idea was to start out with 100 in 2012, and that's where we are right there, that first year of that evaluation. Then in 2013, which we just completed a year, we decided we took 100 of those and whittled them down to 10. We planted those 10. Now from those 10, we need to choose 3 for next year. And then after next year, then we'll choose 1 for 2015, as you see down in the red. And that's what the whole goal of this project is to do that, is to get some seed that is to find it and adapt it and to get it in the hands of the farmer. Okay, so as an example, just to kind of put it in perspective for you, because I mean I work on this stuff all the time and I know I talk about it, you don't know exactly what I always mean when I say small plots. Small plots to some of you, especially the guy from Montana, is probably 100 acres. The guy from Rugby is probably 150 acres. I don't know. Small plot to me is 100 square feet. Okay, and so that's what we started off with. We planted a row, 4-foot row that first year, and then in 2013 we were able to plant larger plots. In this case, the plot is actually the size I take most of my replicated data on, but these are seed increase plots. So basically, our drill does 7 rows, and those are 25 feet long. So that's what we were able to plant from that increase in 2012 is down to one of these. The interesting thing I want to point out, and we'll get into this a little bit more, but actually with Einkorn, I don't know why, doing everything equal, we're actually getting twice the yields of Einkorn that we are Emmer and Spelt, so we're able to plant twice the area. In other words, if I was harvesting 100 grams of Emmer, I'm harvesting 200 grams of Einkorn. If I harvested this year 1,000 grams of Emmer, I'm harvesting 2,000 grams of Einkorn. So I don't know why did we select good Einkorns. We'll find out. That's why we do this work. One of these is considered what I consider a plot. We have to break in their point so we don't get mixed up. So essentially with Emmer, we were planting one of these. In Emmer and Spelt, we're planting one of these. Einkorn, we're planting twice our area. So that's encouraging. We're moving faster with the Einkorns than we thought. Now, maybe we'll have a failure in 2013. We don't know. Or 14, I guess that's coming up. 13 was a good year. So basically, this is what we'll do now. Going into 2014, we will look to be planting strips, again, using that same little drill, but they'll be 100 to 200 feet long. So I just wanted to put that in perspective, in a visual for you to see where we're going with this.
Okay, let's just look at a possible scenario here. Now I made this slide up when we started this. So this is dangerous because this is going to be around until we finish in 16. So far, I feel good. I hope I continue to be right. Again, when we started in 2012, I told you we planted 1 gram, a 4-foot row. You saw that picture. We harvested 100 grams. Well, okay, this is a scenario. Say if we harvested 100 grams. Then in 2013, we took that same 100 grams. We planted that much of an acre. Nobody plants that much of an acre except in your garden, right? And we harvested, like say we harvested 2,500 grams, okay? Again, these are just scenario numbers. So then this 2,500 grams, then now I'm getting close to a 1/10 of an acre. It's a 20th of an acre, actually. I'll plant 5.5 pounds, hopefully harvest 100 pounds. My scenario, I'm using 2,000 pound per acre yields, okay? So now we get to 2015, if I plant this 100 pounds on an acre, I'm gonna have 2,000 pounds. So at the end, we're meeting our goals. So then in 2016, it's gonna be possible to plant 10 to 20 acres, depending upon the seeding rate we wanna use, to whereas when we harvest, we could have 40,000 pounds from some of this seed that we planted 1 gram back in 2012 by hand. This is actually a field of North Dakota common emmer in Lewis Sibbles up in Cathay, North Dakota. In 2008, that's a 100-acre field. That field went 2,000 pounds, actually 1,972 pounds per acre, but 2,000 pounds is close enough, right? And if you look at that, that's a clean field. Lewis had really good production that year. The bottom line, the reason why I show you that is when we started a project without even really knowing where we were going, increasing that small plot, it only took a few years to get to there. So I use this to show you it's real. We can do this if you're willing to work on small scale and if you have partners to help you do this. Obviously, my drill can't do that. I can't plant that by hand. That's the participatory work that we're dealing with, where we've combined with some work with the university, we combine work with farmers. So basically what I'm trying to say, all of us working together, we can meet goals. So this is the einkorn. If you look at these, okay, this is again where we got it from Grin. What they do is they catalog them by this PI number here. So this is a number they give. And actually the lower the number, the longer ago it came in there, because they kind of go in sequence. And then it kind of, and I don't even know what this exactly means, but it's like a name. Sometimes you'll see a name that this gets by. And then it shows you the country, where it came from, and then the state within the country, if there is. Just a little background information to show you where it comes from. So now if you look at, this is some data that we gathered, observational data, because again, we don't have replication, so we can't run statistics on this. But this is just some data to help us try to identify the varieties that we want to pick, or the cultivars and the lines. Early vigor is just as simply a score of going out there early on after emergence and getting it a 1 to 5, 5 being best. Generally when you see our numbers, usually the higher numbers are always the best. You know, so it's a subjective thing. So in other words, you went and you looked at everything and you said, that's the poorest, that's a 1. You went and looked at everything and you said, that's the best, that's a 5. What it does is it gives you a very quick snapshot that day without taking a lot of detailed data by just an observational note to tell which one numerically was better by early season vigor. That's an important character. So basically, you can just look at it. This is just how they ranked. But you see, we didn't really have much differences. There's 6 days difference between these 10. Height is important. We definitely want straw, and so height becomes an important factor, particularly sustainable organic biological farmers like straw better than the 40-foot head combines that don't want the straw because you got to get through there. So straw is an important thing. Whether that stands up or not is important. So that's where we measure lodging. Now in this case, it's the opposite way. Scientists do it different. 0 is best, 9 is worst. So whenever you see lodging scores, this is something that's been done for years. It doesn't even make sense. It should be 1 to 10 instead of 0 to 9. They chose 0 to 9. 0 is standing straight up, 9 is flat.
So would a 5 be that it's halfway falling over, or is it 50% of the plant population?
No, it's basically, think of it as 0, 9. So 5 would be, yeah, 5 is getting pretty hard to pick up.
But then if you look at a population and some are here, you're sort of an average of the entire row of.
Yep, that's what you try to do too, because you're right. You'll have, again, it's a small plot. And there's a lot of variability within that small plot even there. But I mean, you can have one end of the where it fell down a little more than the other end, and you gotta take it into account the best you can, you know. So all scores doesn't mean everybody's gonna score it the same. But what you hope is that one person scored the whole study, so it's all relative. But yeah, good question. Actually, I like the idea that we would go more on percentages than the 0 to 9. But 0 to 9 has been done in the scientific community forever. Okay, and then the yield. This is the amount of seed that we harvested from this area. And so that's where we have on the einkorn. And if you look, we do range from what, 2,600 up to 4,200. So we're seeing some pretty big spreads already because everything was planted all in the same area. This is all equal. It's not replicated because we don't have that much yet, but it's still, this is what we're going to base our numbers off of. And I'm kind of guessing the majority of people are going to pick this one. You know, I was looking over it myself a little bit, and you know, it starts to become hard. You want to keep more, but you can't. And so sometimes, if we tend to get a lot of them from all the same country, you know, if we said, oh, we want all three, you know, I would tend to, I'm not trying to make your picks, but I want you to think about diversity. And so would we want to pick 3 of them from Turkey? Maybe not. Don't know. But no, I think the einkorn is really exciting. So then we had the same thing with Emmer. But interestingly enough, now we were doing everything the same. Look at those yield differences. I mean, we're talking half the seed already after just 2 years. We might have to build some einkorn bins, I'm not sure. But, so we look at here, now we have, Emmer's a little bit unique now. And I haven't quite been able to put a good handle on this, but I can explain it probably briefly. If you notice anything that comes from India, which we picked out 4 of them, the Emmers are a little different. This is where heading date comes in a little bit more important. They tend to be earlier. Now everything was skewed a little bit this year because of our late spring forced heading. Probably don't see as big, you don't see the spread amongst cultivars when you plant later. And so, but all the material from India is early and it's short. It might be too short for some of us because I've seen some of this material, particularly this line Debra, been working with this one for a while. Where is it here someplace? Debra, where is it? Oh, right there, thank you. You know, there's been times when that one there, I've seen it when it's got really stressed, I'm only getting about 12 to 14 inches tall. And that could be hard to pick up. But I'm thinking, and I don't know enough about these early types from India yet, but they're a little different. But I'm thinking these might be good lines for the heavy, high-yielding areas, like out in the East Coast and stuff like that, where these Emmers that don't lodge out in Doug's country, but that go flat when you start getting into, you know, better conditions. These may be better varieties for them. We don't know this yet, but if you'll notice, they're all early and short, and it means they're short, they don't tend to lodge as bad either. Obviously, some of them have some pretty good yields, and the seed does look like pretty good quality. And some of them, to a lesser degree, more than others, actually free thrash, like the Debra. And this is kind of interesting, I'd like to pass it around. This is how I thrashed it. I did not do any dehulling on this. This is Debra, this is Emmer. This is what we're dealing with on the Emmers. And again, you know, we see some pretty big spreads. I think for the most part, I would have to say, if you notice this line here, Yaroslav, there's a very good chance that most of the genetic material that we're working with, whether it's North Dakota Common or any of these land races that ended up in this country, are very similar to this background. Because this is a line that comes out of Russia. And back in the early 1900s, the USDA government brought a lot of that seed over here and did experiments with it to see how this crop was adapted. So a lot of our land-raised material in the Dakotas probably has this background in it.
Variety we've been planting with Emmer is Lucille.
Lucille.
Out of MSU, I think.
Yep.
Are any of these close to that or anything like it?
Actually, I would say Yaroslav is going to be fairly close to Lucille. You know where Lucille came from?
I don't.
Gilstolnik. Got it from Green. in my whole series of things, because I'm kind of a curiosity person, so when I first started getting some of these lines of Grin, I also requested the line that Gil started with and released back in, he first probably pulled that out back in the 80s, early 80s, and worked on it. Well, being a curiosity person and making sure that it's the same thing, I requested the same thing back and grew it for a few years to put it in replicated trials. Yields exactly the same, can't tell the difference. I figured it was the same, but I thought, did it change? But I don't think it did, you know. So yes, Gil, that is where Lucille came from. It's actually PI 6035 or something like that. I can give you the number if you want it, but it actually, that's what he did. He went through Grin and selected the best one and ended up making that. That's where Lucille came from.
Are these replicated trials?
No, they're not, because of that small amount of seed. Now, this is something that I want to pose to the group. What I would like to see is, particularly, I don't think we'll be able to do it with Emmer because there's not enough seed. But I would really like, if this is one of them that we pick, I'd like to put them in a replicated trial this year. But a replicated trial, even though I'm planting small areas, it takes me a pound of seed. So if I plant one trial, and I usually like to do 3 sites. So far I've been planting 3 sites for these crops. Well, that means I've used 3 pounds of seed. So to get replicated data, we would be giving up 30% of our seed. Because when you do replicated plots, you don't save the seed back because it's too hard to do. Because you're running, you know, the way we do it, the combine runs through. Even though these little combines clean out well, they're not pure, you know. And so it's really hard to mix. And I know these guys, I've been going over it with our team here a lot. It's hard to do a variety trial and save seed from it. It's really hard to do. So I tend to try to, I mean, there are times when you'll do that, but it takes you a lot longer. And then what you do is you need to kind of be able to get in and get them all cleaned out of that same one and then clean out the combine again. And then make sure when it gets to the cleaner that same thing happens. You know? So yeah, it's hard to do both. And these are not replicated. There's just a single observation.
What's the protein testing?
Protein testing. We haven't done that yet. What will happen is now that we can start doing some dehulling, then we can start running some proteins on it, because you can't send it to any lab and say run a protein on it because they don't have a dehuller. And there are, and common proteins are run on wheat and many other crops now by near infrared, so you don't have to destroy the seed. There are no calibrations for these crops yet. So in order to do proteins, you need to first, number one, get them dehulled, send them into a lab, then they will run wet chemistry on them. There are no calibration curves yet. And wet chemistry is more precise, takes longer, costs more money. But this year, I've not done any, I won't do any proteins. Next year, the goal is to start running some proteins on some of the lines that come out of the variety trials. Myself, like I said, I've never ran a protein yet because I've never had a place to do it. And I know I've seen some people run them through NIRS machines, but I don't know what good the number means when there's no calibrations. There is none. And that just to help you understand how, you know how they develop calibrations for these machines? You send a lot of samples through wet chemistry, you know, where that's absolute, and then you use those to calibrate the machine. So I think it's something like 1,000 or 1,000 to 3,000 samples makes up the calibration. But good point. Proteins, that's the next step. You know, a lot of the work that's been done with these ancient grains on the scientific level and other things is, you know, it's been mainly to first test adaptation, see how the crop grows, and then, you know, and to get some data on it, because, you know, that's why we're here today, because we found we can grow it. I'm not so sure we can dehull it. I think there's a few people that are gonna raise their hand on that one, you know. And so, you know, we're at the next juncture. We've proved, it's, you know, the point is it's been proven it can be raised. And it's proven that people want to buy it. But the people that are dehulling it, their machines have to run slower, they're losing money. The people that are buying it say, come on, you can dehull it, we can do it, because they have the sales. So we're all in this together. And yeah, so The next steps are some proteins, and I think it really to go way further, and Frank will be able to discuss this much better than me, but to start running, and which we are going to a certain extent on Einkorn, run different amino acids and different, different components of these, because they're unique. They have different micronutrient levels and stuff too. The literature shows it. We don't know on the particular lines we're growing what they all are, but I think that's why there's interest, because they probably are more nutrient dense. These are the 3 sites that I had Emmer variety trials on there this year. It was Carrington at the research farm I work on, Robinson, Ann Ongsted's farm, Cathay, Louis Seibel's farm. Really nice when you have all the same treatments and you can make averages. It makes it so much easier. But basically we had 15 lines of Emmer here. Interesting thing about it is I thought that when I first combined them, Robinson was very good test weight, some of the highest quality I've ever raised. I thought this is going to dehull better than anything. It didn't. It was essentially equal. Cathay did dehull quite a bit lower. If you look at the mean, 55 compared to 60 compared to 61. This was a very late planting date. It was so wet there I could not see till June 7th. And we still had pretty good yield, and particularly on Emmer as compared to wheat. Emmer really did good. And we still had 2,000 pound Emmer and 38 pound test weight. But it didn't dehull very well. So there was a difference there. Now, again, what I did, let's see. The first one that went around, that bigger sample, is dehulled North Dakota Common. Basically, what I have here is, this is the hulls that came off the top of my cleaner, off the top of the sieve. There are some that have some grain in it and stuff too. This is what come off the bottom, the little bits and pieces in the bottom. So you can see this is the components off the 2 ends of the cleaners from it when it's dehulled. So basically, what I did was weighed everything going in, dehulled it, cleaned it, weighed it coming back, and you can see that I was only getting 60%. When you look at the old literature, or when you do it by hand, when you do it by hand and you get everyone out of there, it's about 20% of that yield is hulls. But I don't know if there's machines that can do that. So in this case, that machine did 60%. So that's where we're at. Now I don't have the data presented for Einkorn, but I dehulled lower amounts on the Einkorn. Einkorn didn't pop out near as good as the Emmer did. So this is just the first data that we're looking at. Oh, I'm sorry. This would be then the hulled version. Now you'll notice the equipment I had to use wasn't 100% perfect, but the best I went with. There are a few in here that I couldn't separate out that have a hull on them yet. Still, everything will be uniform and equal, but this is how that would look. What I'm going to intend to do next year is do the same procedure, but I want to use a dockage tester to run the sample over so I can separate out the components better.
I just want to make sure I understand. You're saying 60 and 55% coming off the combine was dehulled?
Yes. In other words, Doug, like say that one big sample that he has there. Okay. When I put this, I weighed this going into the dehuller. So this is what come off the combine, and I dehulled it and then cleaned it back, and then weighed, that portion there, which is the grain. Basically, I was getting 60% of the weight that went into before I hulled it. So in other words, from this off the combine, you're only getting 60% of the kernels. You follow me?
After commercial or
After cleaning it. Yep. Now I know commercial equipment's going to be better, but what I'm trying to do is first get some baseline data to distinguish between the varieties. Because here's what I'm going to say right away: if dehulling is that much of a problem, and if I don't even know why we want to plant grade 4 anymore. If it only does 43%, it's that much lower. Or look at these 2 PIs. PI 148. Go on and get 27% of it out of there. What do we want to grow that for? We can't grow that. So I'm thinking we need to start throwing some of these out. Fortunately, Lucille is one of the higher ones. But now look at these. These numbers are a little bit off, but it's still relative. I need to keep in the trial. Look at Debra. Basically, there was only 4% that didn't dehull, whether it's on the combine or through the dehuller. Now Kamut, it's in the trial, but I still needed to do it for numbers. Kamut actually dehulls itself too, so that's why you see these are so much higher. But basically when you start looking at some of these, the best thing that we have going for us is the lines that we are growing. Lucille is higher, North Dakota Emmer, North Dakota Common is up there. And even commercially, Red Vernal can be going, because I know there's that can be a commercial variety. There's some farmers that got a seed of that. I don't know. That one looks pretty good on the dehulling and on the yield. So that's where we're at. It's a learning curve on this dehulling, but that's that first data we gathered. And one of the things I think is important on the research side is that if we can start screening some of these lines, I mean, even though they might yield 4,000 pounds the acre, if we can only get 20% of it out, I don't think anybody wants that. So I think that's an important thing, to be able to start screening some of these for that. And I know a strong interest that we have, and in particular that what Frank would have, where this would be important for him if he wanted to start making some crosses. There's great discussion and just some of the first preliminary data that's been presented and stuff. So like I said, it's a learning curve. Hopefully that we'll all figure out how to do this together and be able to work with research, farmers, and industry to solve this problem, because I think together we can do it.